drought, a plant goes long periods without receiving any water. Water moves from the soil to the plant to leaves and escapes through transpiration. With water removal, such as in conditions of drought, the seed enters a stage where metabolism decreases, and it is not reactivated until favorable conditions return. This is achieved through desiccation tolerance, a mechanism that allows for the tolerance of further dehydration of the plant. Without this biological process, the plant will proceed to wilt. Wilting reduces water loss by decreasing the surface area of leaves, leading to a diminished ability to transpire and a decreased growth. Wilting is the process of water moving out of the vacuole, which collapses the cell wall. If a plant goes too long without having any water, it will die.
Now we will be learning about the biological significance of desiccation tolerance. Once again, desiccation tolerance is the plant's ability to rehydrate from almost no water content through a series of metabolic mechanisms. This promotes seed longevity and helps the plant survive with little water and to recover normal function upon rehydration. This also allows organisms to live on dry land and allows organisms to survive extreme water loss without extreme damage. If a plant doesn't adapt to its environment, it will die. In order to prevent this from happening, some solutions to desiccation tolerance include stomata closure, evolutionary plant size changes, late embryogenesis abundant proteins, also known as LEA proteins, the delaying of seed germination, and the accumulation of compatible solutes. First, to be able to grow, plants need to take up water from the soil and carbon dioxide from the atmosphere and use it in photosynthesis. This is done by carbon dioxide uptake through the stomatal pores, which is where the water is. When the stomata are open, carbon dioxide is taken up while water is transpired. On the other hand, when the stomata are closed, little carbon dioxide is taken up and the transpiration levels are lowered. When the environmental conditions are unfavorable, plants can regulate the amount of water loss by opening and closing the stomata and sacrificing carbon dioxide uptake. Moving on, size changes are important because resurrection plants are pretty small. They do not need a lot of water to stay healthy. For example, when the weather is dry, resurrection ferns turn gray and shrivel up. But when the rain comes, resurrection ferns will spring to life within a matter of hours. Another example of this is shown in small cactuses. Pine trees need a lot of material and water, and it is difficult to transport water, versus in small cactuses, not much water is needed to survive. In addition, LEA proteins have been known for many years to accumulate in maturing plant seeds. They are extremely hydrophilic and function to keep plants in embryonic state. They act as molecular chaperones and form cytoskeletal filaments to counteract desiccation stress. Furthermore, seeds can delay germination for decades or even centuries, and they wait until conditions are right. An example of this is the Nelumba nucifera lotus seed from China. Nelumba seeds remain viable for many years, and if the seed is placed into an ideal habitat for growth, it may still remain dormant for many decades before sprouting. Being resistant to sprouting has been shown as a survival advantage in many plants. Lastly, the accumulation of compatible solutes is very beneficial. It increases cellular osmolarity, which leads to an influx of water into the cell and a reduced efflux. The accumulation of compatible solutes is often regarded as a basic strategy for the protection and survival of plants in osmotic stress.
3. How do these biological processes work? During a drought, there are high levels of ABA and that leads to stomata closure. Without a drought, ABA levels are lower and there's going to be no closure. You can think of the stomata as a mouth opening that's on the plant and allows for transpiration to happen. When there is a drought, the ABA levels will accumulate and that leads to binding of the PYR, PYL, and RCAR complex. Then that complex will bind with PP2C, which finally combines with SN RK2 and allows it to be phosphorylated and allow ion channels to open. Chlorine ions and potassium ions will leave the cell, and this process ultimately leads to the closure of the stomata. Calcium will also enter the vacuole via the cytoplasm, and by doing so, this allows the vacuole to expand. Due to its expansion, it will cause the cell wall to expand and remain rigid. This is important to delay the wilting of the plant and to survive a drought. During a drought, when ABA levels are high, this also prevents the inward flux of potassium, which isn't necessary for the stomata closure, but it's important to keep the stomata from opening. Desiccation tolerance also includes size changes. Bryophytes such as moss and liverworts have adapted to change their sizes and become smaller. They have constitutive cellular protection and rehydration-inducible recovery to repair cellular damage during rapid desiccation and dehydration. They also have constitutive endocytosis, meaning the proteins in the cell wall, the microfibrils and the microtubules will constantly be degraded and recycled by the lysosome. These plants have a very low metabolic rate as well as slow growth and limited size. A low metabolic rate helps the plant to conserve energy and resources, especially in the case of a drought. Plants will also delay germination. They do so by the interactions of different hormones such as GA and ABA. GA promotes germination and it destroys DELA, which are germination repressors. ABA represses germination by stimulating LEA proteins to keep the plant in an embryonic state. In normal conditions, GA can inhibit DELA, which then does not activate ABA and lead to germination. However, when there is a drought present, GA gets inhibited, which allows for DELA to activate ABA, and then that inhibits germination. It's important to delay germination during a time of drought because if the plant were to germinate during a drought, it would not have the resources that it needs to be able to grow, and it would just dry out and die. Finally, plants will accumulate compatible solutes such as proline, mannitol, glycine, and betaine. When these solutes enter the vacuoles, osmolarity takes over and water follows the solutes into the vacuoles, which expand the vacuoles and keeps the cell wall rigid to prevent the plant from wilting. Since water follows areas of high solute concentration, as long as the solutes remain in the vacuole, water will also stay in the vacuole, and that's very important because by retaining water, the plant can maintain its structure and survive longer during a drought. And that's how plants induce the desiccation tolerance.